All right, so we spent the last podcast talking about origin of life, and in particular, not how life could get here, but how could simply those little building blocks of life get here. The organic monomers, the amino acids, the nucleotides, because again, they certainly cannot spontaneously form by themselves on today's Earth. But it has been hypothesized that perhaps they could have spontaneously formed on the early Earth from just the most simple inorganic precursors if the conditions had been correct. So let's just suppose, okay, so we've got organic monomers. Now what? What's the next step? Okay. Remember these steps towards life from non-life as it is discussed by scientists possibly went in an order something like this. And what we spent last time talking about was this critical step, getting organics from inorganics. But now what do we do with these building blocks? Well, we need to put them together into bigger building blocks. We need to make not just monomers, but complex organic polymers. And the question is, well, how could that have happened? Not life. These organic polymers, remember, are not living things yet. They're just bigger, more complicated organic compounds. Like instead of a single amino acid, maybe we're talking about a polypeptide. Instead of a single nucleotide, we're talking about starting to form strands of nucleotides that we would call nucleic acids and things like that. So how could these form without a living thing with enzymes and things to put them together like what we have now with life all over the planet? Remember, there was no life with little enzymes assembling these things all the time. How could they have just come together? Bottom line is we don't know. But again, there are hypotheses that exist that are at least to some degree testable, to at least determine if they could be possible or not. It has been determined that spontaneous formation of polymers, and I should write of polymers, of these organic polymers, is possible on certain catalytic surfaces, of which certain clays that might have been present on the early Earth can act as these catalysts. Again, hydrothermal vents with all of their heat and the rich chemical soup that surrounds them have also been thought to possibly be hospitable places where organic monomers could have joined up into making these organic polymers. Today, we don't get the formation of these polymers without enzymes. And remember, enzymes would not have existed at the early Earth. They're too complex. So we would need some kind of inorganic catalyst, and the hydrothermal vents, or perhaps again certain clays on the surface of the Earth, could have acted as those catalysts for formation. So let's suppose we have our polymers. What's next? Well, we might need to package these polymers into something that started to maybe look somewhat cell-like. We need a package, a bubble to keep these organic molecules in. Why? Because this packaging would allow us to start to create something different from the general environment. A separate entity from the general environment. It would allow this inside compartment to actually become different, at least in small ways, from the outside of the compartment. If we have that, then collection of certain materials could occur within this little pre-cell type structure. And this would be kind of the beginnings of metabolism, collecting certain materials that are needed or to be used to create certain molecules. Reproduction of the structure might then be able to occur once we can be accumulating materials we need and so on. And if we can get some reproduction, then perhaps we can start talking about heredity, or certainly that is the essence of reproduction. So what would these little earliest packages of organic compounds look like? They've been given a name, actually several kind of names that are kind of used to talk about these. Protobiont is one, protocell is another. At any rate, these are collections. These protobionts would be collections of abiotically made, in other words, not made by a living thing, molecules, but they are surrounded by a membrane, okay? And they exhibit at least some, not all, but some of the properties associated with life. These protobionts can carry out simple metabolic activities, these sorting, collecting, and sorting of molecules. Certain things are allowed into the membrane while other things are not. It's a very simple type of metabolism. Simple reproduction, where the little spheres will break apart and create two spheres where there was only one. And again, as we mentioned, they can somewhat maintain an internal environment that is different from the external one, so that just like your environment is different from what's outside of you, we are starting to get this creation of something that is different from just a sack of chemicals exactly like what's outside the protobiont. So what evidence is there, okay, for these protobionts? Again, there's really no fossil evidence, but we do know that small membrane-bound droplets do form all by themselves spontaneously when lipids are added to water, just because of the chemistry of the lipids in the water. Such structures are called liposomes. Okay, and we see them form today. We do it in the lab, whatever. And we know things like liposomes are selectively permeable. They can create inner conditions that are different from the outer conditions. They will let certain things pass through and other things will not pass through the membrane. They can even accumulate charged particles, ions and things, and discharge electrically because of this, very much like a nerve cell does, which we'll be talking about more soon. There is evidence for these protocells, protobionts. We can kind of create them in a lab situation. Does that mean that's what happened? No. Again, nobody was there to write it down, but we can test a hypothesis to see if it was possible. Which came first, metabolism or heredity? It's a really important question, because once we've got these packets, if we want them to persist in the environment, of course they have to be able to maintain themselves, and that's kind of metabolic activity. But if we want them to pass into next generations, which is kind of the essence of what life does, we've got to have heredity. So metabolism, just be aware, is these sets of chemical reactions, often very complex sets of reactions, that allow life to collect molecules, process energy, to grow, all those things. And these are, in today's living things, all catalyzed by enzymes. These reactions don't go if there aren't enzymes present for them. Heredity, on the other hand, is the process that allows life forms to pass the instructions for these life functions on to their offspring, so that their offspring can do these too. The offspring don't have to invent these processes on their own. The instructions are passed on to them, and they can begin doing them right away. Do remember that natural selection, heredity is a requirement for natural selection. So before we can have natural selection acting on, say, hey, this is a successful process, this is going to continue to go on, this is an unsuccessful process, this is not going to be allowed to go on, that can't happen unless there's this hereditary component where we can see who passes on the successful traits to the most offspring and all of that. the bottom line to which of these processes came first is we don't know. There are two schools of thought, obviously, and a third school of thought that suggests, well, probably these things were happening together at the same time. But it's a very important piece of the puzzle, and how these two processes could have come to exist is an incredibly important evolutionary question. Now, if we are talking heredity, what would have been the first hereditary material? We all know that DNA is very famous, and what is the main hereditary molecule now? But probably it was not originally. It was probably RNA, because RNA is a lot more versatile than DNA. DNA, if you'll recall, is this very locked-in, ladder-shaped molecule. And it is not really prone, it will mutate, but it's very well protected. One side of the ladder protects the other side. I don't know if you remember that for every A, there is always a T. For every G, there's always a C. If you can recall that, we'll talk much more about that later. But if this is an A, we know this is a T. If this is a G, we know this is a C. The compounds on each side of the ladder, and they lock together, and the molecule is remarkably stable, is the point. RNA is not quite so stable. It is a single-stranded molecule, and as such, these bases that these A's, T's, C's, and G's on the RNA, actually there's no T's, there's U's, but we'll talk about that later too, are more open to the environment, and indeed they're more open to interacting with each other. This thing can bend and fold and look strikingly like a protein in the complexity of its shape. And indeed, because it can do that, whereas DNA can't, DNA is the double helix and that's it. RNA can shape itself in really amazing ways and actually can act as a catalyst. Proteins aren't the only catalyst, and in fact it is thought that RNA could in fact catalyze its own creation, its own synthesis, its own replication. And that's important because there would have been no need for separate protein enzymes to be present initially. RNA could have done its own thing by itself. RNA that acts like an enzyme is called a ribozyme. So if you ever see that word, that's all it's talking about, is RNA that can act like an enzyme. So leaning towards RNA being the first genetic material because of its versatility, its ability to catalyze its own chemistry, and so on. But again, we don't know. That is speculation, though educated speculation. So, remember, we are talking about steps to getting to life. So we spent podcast 1 going to that first step. In this podcast, we've gone from monomers to polymers, from polymers to these protocells or protobionts, from these protocells to getting our complex molecular processes, whether that's heredity, don't forget metabolism, we've got to have that too. And then, once we have got all of that, then we are talking life, the very first cells, okay? So, that's where we're headed next. My little things are not... There we go. Once the cells are established on Earth, then what? Then we're off to the races. So we're going to touch base on that real quick, but that's the end of the second Origin of Life podcast, and I will see you in the next one. Thank you for watching. Take care. See you in the next podcast.